This video is going to cover glucose metabolism. Now, glucose metabolism is a big topic, so for this short video we're going to focus solely on glycolysis. So the learning outcome for today's video is to explain the processes involved in carbohydrate metabolism in a healthy individual. And I'm going to talk about glucose transporters and glycolysis as mentioned. This is an overview of the metabolic pathways that I'm going to talk you through in a number of videos. For today's video, as mentioned, we're talking about carbohydrates, which is in green, particularly glucose, and we're going to go through glycolysis, so getting from that top carbohydrate box through glucose 6-phosphate, down the arrow through glycolysis to pyruvate. So the focus of today's lecture will be this. I'll use this as a reference point for a number of my metabolic videos so that you can see how everything fits together once you've viewed them all. So if we start by having a look at glucose. Glucose, in order to provide a fuel for the cell, needs to be taken up into cells. Now, it gets through the cell membrane by something called a glucose transporter. There's a number of different types of glucose transporters, but all of them begin with GLUT, so GLUT for glucose transporters. Now in this short animation here you can see that the glucose is shown as the purple hexagonals and sodium is the small yellow dots. Now in order to get glucose from the outside of the cell inside the cell, we need to transport it across its gradient, meaning we want to go from a low concentration of glucose to a higher concentration inside the cell. And that's why we need the transporter. To do this, we rely on the sodium gradient, which is higher outside the cell than inside the cell. So when you have glucose that you need to get inside the cell, the glucose and some sodium attaches to the glucose transporter. The transporter then changes conformation, where it then allows that sodium and glucose to be released inside the cytosol of the cell, as shown. So that's how we get glucose from a low concentration area to a high concentration area through the glucose transporter. There are a number of different types of glucose transporters in the cell. Now these have different numbers, GLUT 1, 2, 3 and 4, and each of them operates slightly differently, and that's relative to the cell type that they're part of. So for example, the GLUT1 transporter is in fetal tissue and some adult cells. Now these allow low-level glucose uptake all the time. So these cell types always require a small amount of glucose to operate, and the GLUT1 transporters enable those cells to access glucose at all times. The GLUT2 receptors are very important. They're found on liver cells, pancreatic beta cells, and some other cells listed here. Now the important thing about GLUT2 transporters is that they allow the transport of glucose in both directions. So they take glucose into the cell, but they also put glucose from inside the cell back out into the bloodstream. Now the liver is the metabolic control center of the body, and it's important that those liver cells are able to put glucose back out into the bloodstream, because it's the liver that undergoes a metabolic process called gluconeogenesis, which we'll talk about in another video. But gluconeogenesis is the process of creating glucose. So those GLUT2 receptors transporters are important in those cells. GLUT3 transporters are important for neurons in the placenta because they allow transport even when we have a low glucose concentration. So you can imagine a placenta and our nervous system requires energy at all times, irrespective of how much glucose is in our blood at the time. And finally, on this slide, the GLUT4 receptors I want to draw your attention to, because these are the ones that are highly regulated. So they respond to the amount of blood glucose, and they do that by insulin regulation. So adipose cells and muscle cells only take up glucose when the body is in a state that allows that glucose to be used for muscle energy, for storage in adipose cells and storage in the muscles. So they're highly regulated, and that's done through the GLUT4 transporters. So glycolysis is common in all cells. So any cell can carry out glycolysis, and importantly, it does not require oxygen, which means it can occur under anaerobic conditions, which is important. Now, the location of glycolysis is actually in the cytosol of the cell. So if we have a look in your image here, you've got a cell depicted. Now, glucose comes out of the bloodstream and crosses the cell membrane through the glucose transporter into the cytosol of the cell. So glycolysis happens in the cytosol. Once glycolysis has produced pyruvate, and I will show you how that happens in a minute, the remaining steps to get the greatest amount of energy from that pyruvate involve the mitochondria. But for this video, we're talking about glycolysis, and that's occurring in the cytosol of the cell. So what is glycolysis? It is a sequence of reactions, and it takes one molecule of glucose to create 2 molecules of pyruvate. And it has a net production of 2 ATP molecules. And I'll go through that in more detail in a second. But this is the overview of what you get. You start with with glucose undergoing glycolysis to produce 2 molecules of pyruvate. From there, the pyruvate can either be further metabolized into lactate if you are in an anaerobic environment still. It can be completely oxidized to carbon dioxide and water if you are in an aerobic environment, meaning if you have oxygen. And other organisms, not humans, but different bacteria, can actually metabolize pyruvate to ethanol. Yeasts can do that too, and that's a process called fermentation. So here is glycolysis step by step. Now I'm going to highlight the important bits, but I think it's worth your while pausing on this for a moment to see what all the different reactions are. Now this is the type of thing that I want you to understand the processes of glycolysis and the purposes of glycolysis, rather than memorizing a whole number of different molecular names without actually knowing what's happening. So let's go through this step by step. You start with glucose, and we've got that inside the cell now, so it's come through the glucose transporter. The very first step of glycolysis actually uses up energy, so you need some ATP to convert that glucose into glucose 6-phosphate. Now that's an important step because glucose 6-phosphate can't then leave the cell. It's a molecule that must stay inside the cell, and there's no way for it to leave the cell. So in a way, you've now trapped what was glucose inside the cell. There are then a few more reactions that occur with enzymes, and a second ATP is used in that first stage of glycolysis. So stage 1 of glycolysis consumes 2 ATP molecules, and therefore that uses energy. You then end up at the end of stage 1 with 2 glyceraldehyde 3-phosphate or G3P molecules. So you've taken a 6-carbon glucose and broken it down into 2 3-carbon molecules. And then we proceed into stage 2 of glycolysis. Now in the second stage of glycolysis, this is where you're actually generating energy. Now I'm not going to go through all the names of the compounds and enzymes, but what I think is important for you to understand is that this stage actually creates energy. So we've got those 2 lots of 3-carbon molecules, and as it proceeds through the second stage, which you can pause and read the little notes here, but importantly, it produces 4 ATP from those 2 3-carbon molecules. And when I say that, I mean you've got 2 lots of a 3-carbon molecule. At the end of glycolysis, you have pyruvate, and you've got 2 pyruvates because pyruvate is a 3-carbon molecule again. So you started with glucose, which had 6 carbons, and you've ended with 2 pyruvates, each with 3 carbons. You've also gained overall 2 ATP. So there's a net production of 2 ATP. So you have had 2 ATP consumed in stage 1, 4 produced in stage 2, and therefore you have a net production of 2 ATP. So looking in more detail, stage 1 here has listed the names of the enzymes. The most important one there, I think, for you to know about is hexokinase. Now, depending on the cell type, sometimes that might be called glucokinase, but that is the enzyme that converts glucose to glucose 6-phosphate and actually therefore traps glucose 6-phosphate inside the cell. So the glucose can't leave the cell again. Until that reaction has occurred, the glucose could then go across the glucose gradient and go from inside the cell back to outside the cell. So you've gone down from these ring structures, 6 carbons in the ring, and ultimately at the end of this stage you end up with a 3-carbon molecule, like I said, glyceraldehyde 3-phosphate. And I'm showing you this because in a later video we will come back to these molecules, but in that video we'll talk about gluconeogenesis, where we're actually trying to create glucose rather than break glucose down. And glyceraldehyde 3-phosphate is an important entry point for a number of substrates. So as I said, the important point from this that I want you to get out of it is that glucose 6-phosphate cannot pass back through the membrane, whereas glucose can. The other important thing of this stage is that there's a consumption of 2 ATP. So it's an energy consuming stage. So as I said, 2 ATP are consumed. The second stage, as I mentioned, you've started with the G3P and their 3 carbon molecules, and as you go through this stage, from every 1 glucose molecule you will go through this stage twice, because you had 6 carbons and then you break it down into 2 lots of 3, so each of those 3 carbon molecules will go through this stage to ultimately produce pyruvate. Now the important thing about this stage is that it produces ATP. So as mentioned, there's a net production of 2 ATP per reactions shown on the screen, but we go through that twice. So overall, there's 4 ATP produced. So once we have pyruvate, there are a number of fates of pyruvate. As mentioned, we as humans can't undergo all of these reactions. So pyruvate's fermentation to ethanol only occurs in microorganisms and yeasts. So we cannot do that in our body. There is another video I'll do which will focus on how we get pyruvate to lactate, and that's called, and then recycle that, and that's called the Cori cycle. That occurs in muscle cells once the muscle cells have used up all their oxygen. So that's during anaerobic conditions. But importantly, I want you to remember that as we move around and use most muscles, and even when we begin exercising, for the most part, our pyruvate is going to go down the aerobic pathway, which is the complete oxidation to carbon dioxide and water to produce acetyl-CoA. So as we go through different energy stages, initially we would always prefer to make energy using oxygen because we will yield a lot more energy that way, which I'll show you once we get to those videos on the Krebs cycle and the electron transport chain. So there are these 3 fates of pyruvate that occur under different conditions and in different organisms. 2 of them is where oxygen is absent, so they're anaerobic conditions. 1 of them requires oxygen to have the complete oxidation of pyruvate. So the summary of this short video is that we need to get glucose into the cell, and we do that via the glucose transporters, and there are a number of different glucose transporters that belong to different cell types. Glycolysis occurs in the cytosol, and it occurs under anaerobic conditions, so you don't require oxygen for that to happen. There's a net production of 2 ATP per glucose molecule, and we know that glycolysis converts 1 glucose into 2 pyruvates. Now, under anaerobic condition, glucose in the muscle is metabolized by the Cori cycle, which I'll show you in a different video. So as always, if you have any questions, please don't hesitate to contact me. But importantly, there are some readings here that could support your learning if you're finding this topic challenging. And I've put a link down here, which you'll have to type into your browser. But importantly, there's some animations in chapters 14 and 15 that might help you understand glycolysis if you'd like to go over it. Thank you for listening.